As we continue with the prokaryotes, again, there are a few other organisms that are discussed in that chapter. Again, many of these are not particular organisms that are going to cause human disease, but just helps you to identify some of the relationships that exist between the environment and the multiple species that are there. Theobacillus is an organism. It's a species. It's a sulfur oxidizing bacteria, important in the sulfur cycle. Spirillum voltans, relatively large gram-negative anaerobic bacteria. Usually see it exist in fresh water. Many of the spirillums we'll talk about in some other diseases as well. You don't have to know about that. Now, Burkhalter's disease, this is one that's important for you to recognize. This is of the genus Pseudomonas. We'll see Pseudomonas a lot in diseases that cause problems in humans. It's an aerobic gram-negative rod. Motile has a single polar flagellum. The turf, I believe pseudomonads were one of the assignments for you on your lab, not this last week, but I think previous labs. I think pseudomonads are listed there. Nutritionally, they're capable of degrading a wide spectrum of organic molecules. Again, you can see why the human body may be a great niche for this organism to grow because we are organic creatures. Bordetella, another important one because it leads to a condition referred to as pertussis. You should be familiar with. Pertussis relates to whooping cough, prevalent disease in children. Neisseria, another organism that's important to recognize. This is a gram-negative diplococci, gram-negative cocci. Neisseria gonorrhea causes gonorrhea. Neisseria meningitidis, meningococcal meningitis. Very important disease to be able to recognize and to be able to identify. Neisseria. So glia, you don't really have to worry about that. This is just some illustrations, pictures out of your textbook that help to identify particular organisms. Theobacillus chemoautotrope, again, involved in the sulfur cycle. Again, I'm not going to hold you for recognizing that. I do want you to be able to recognize these organisms, though. You should be able to recognize Neisseria, Neisseria meningitidis, Neisseria gonorrhea, Spirillum. Again, these organisms have particular characteristics. Fembri, the diplococci of Neisseria gonorrhea, is very pathognomonic and should be easily recognizable. Spirillum, if you remember when we talked about the motility of particular organisms and the arrangements of the flagella in these organisms. Bordetella, again, leads to the disease pertussis. Burkhalter, related to Pseudomonas, causes significant infections inside hospitals. Nosocomial infection, if you don't know that definition, make sure you know the definition of nosocomial infections. Nosocomial infections are infections that are typically derived from hospitals and our clinical settings. Nosocomial infections are very difficult to treat because the bacteria actually comes from those environments through which you would see antibiotic therapy, antibiotic treatment, so they have mutated and now typically have developed resistance to various types of organisms. Burgata, you don't have to know. Another organism, Franciella, not an uncommon disease that we see that affects humans. Tularemia is actually one of the diseases that we study when we think about bioterrorism types of agents because of its ability to be spread to the human population. The other name for it is rabbit fever. You should be familiar with that. Franciella, tularemia. Another name, rabbit fever. Bacterial disease associated with both animals and in humans. Many wild and domestic animals can also be infected, but the rabbit most often is involved in disease outbreaks. Pseudomonas, I've already talked about, particularly in the hospital, is Pseudomonas aeruginosus. It actually produces a blue-green pigment, can affect the urinary tract. Very common in patients who suffer from burns. Very common in burn patients. So we see burn wounds, and burn victims suffer from Pseudomonas infections. Pseudomonads are common in the soil. They could grow in refrigerated temperatures. They're also able to decompose chemicals such as pesticides in the soil. Ability to grow in aseptic solutions. key point, ability to grow in aseptic solutions. We also may see it grow in whirlpool baths, where we also may see it grow in resistant antibiotics as well. This makes the organism pretty troublesome, very difficult to eradicate it. Many pseudomonads basically substitute nitrates for oxygen during anaerobic respiration, depleting nitrate fertilizers. There are other pseudomonads in the environment as well. As you can see, there's the syringe is basically a pseudomonad that's a plant pathogen. I'm not going to ask you about that. Pseudomonas aeruginosus, as you saw on the previous slide, is the one that typically we see that infects humans, either urinary tract or in burn patients. Azobacter, Azodomonas, basically are encapsulated organisms that we see in the environment. Moraxella, this is an organism that can infect the eye of humans. Another organism it's important to know and be able to recognize. Moraxella can cause these infections inside the eye. Typically it can be treated with topical solutions, topical antibiotic solutions applied there. Leuconata is just a particular family name. Legionella, you probably have heard about in the media before. Legionella disease. Last year, I believe, they suspected an outbreak at the VA hospital, but they later found through cultures and stuff it was not Legionnaire's disease. But Legionella basically is a respiratory type of disease. Legionellosis basically infects the lungs, can have mild to severe cases, and in severe cases it can even lead to death. In the mild form, we see it, the other name referred to as Pontiac fever. And you should know the other name for it, Pontiac fever as well. More severe forms of infection, it leads to a type of pneumonia referred to as Legionnaire's disease. This got its name back in 1976, and I remember this well. Most of you probably don't. But Legionnaire's disease got its name in 1976 because there was an outbreak of pneumonia related to a convention that was going on by the American Legion in Philadelphia. And it caused a great panic because they thought this was going to spread across the country. But it ends up that this particular type of organism grows in moist environments. So it got in the air conditioning system in this particular convention hall. And so people inhaled the microorganism through water mist. People can be exposed through aerosol mist-producing devices, especially water heaters and air conditioning systems in homes, workplaces, hospitals, and public places. It lives in an environment where there are groups of people are exposed to a common water source or a mist. When Dr. Sloan talks to you next week about epidemiology, you'll see how certain diseases can be tracked based upon populations that get infected. And what epidemiologists do is they just get historical information from people. You got sick, tell me the places that you've been to. Well, I went to the airport this week. I was at a convention in Maryland this week. Before that, I was over in South America. And then you find people that have similar symptoms that report it to hospital. You find their travel pattern. No, I haven't been on the airplane, but I was in South America last week. And then you talk to the next person. No, I wasn't on the airplane. No, I haven't been to any conventions. I was in South America 2 weeks ago. And so then you begin to get a pattern of information from individuals, and it helps you to track diseases. Locations, food sources. You know, it's not uncommon that people go to a social event, maybe a state fair, and then a number of different people start reporting to emergency rooms with stomach pain, nausea, and vomiting. And you get the history from each of the individuals. Wow, we saw 3 people today had nausea and vomiting. All 3 people were at the state fair. What happened at University Community Hospital? Wow, they saw 12 people that had been to the state fair. All of those people ate the potato salad. You know, you go to St. Joe's. So you begin to develop a pattern of disease presentations, and then it tracks you back to initial cause. And that's kind of what happened with Legionella disease. The outbreak was basically traced back to a particular area, the air conditioning system in the convention hall. And you can use the same thing to be able to find patterns of diseases. Can be traced back to whirlpools, showers, humidifiers. Read the story of Typhoid Mary. Look it up on the internet, Typhoid Mary, and read the story about Typhoid Mary, how she carried typhus, and she kept being arrested. They put her in prison. she'd go right back to cooking food again, and people continued to get sick because they were exposed to her. So, but epidemiologists were able to follow and track a pattern of disease, and the same mechanism is still used today. Other diseases, Coxiella is another one. It leads to Q fever. You should be familiar with Coxiella burnetii. It resembles rickettsia, resembles mammalian host cells that reproduce. Causes Q fever, which is transmitted through cattle and ticks, and then to humans who contact it, or through aerosol. Can lead to acute febrile diseases, febrile referring to fever, acute febrile diseases, sudden onset of chills, headaches, weakness, malaise, severe sweats, pneumonitis, inflammation in the lining of the lung. You can also see pericarditis, which is inflammation in the lining around the heart. Hepatitis, inflammation in the liver. Hepatitis different from the viral hepatitis of A, B, and C that you may be familiar with. This is generated through infection through Q fever, and then generalized infection, chronic endocarditis, inflammation in the lining of the heart itself. Many people can be asymptomatic, and then there are rare cases, but also can lead to death as well. And this is an illustration of Pseudomonas. Again, this is taken from your textbook. Organism, again, that I would expect you to be able to identify, recognize that Pseudomonas, very common in patients that have burns. So we see it a lot in burn patients. Remember that Pseudomonas will grow in antibiotic solutions. It will grow in antiseptic solutions as well. It's a nitrogen-fixing organism, anaerobic organism as well. It has a polar flagella. So important organism to be able to identify, and I am sure when you work in clinical settings you will get reports of patients that have Pseudomonas infections, particularly Pseudomonas aeruginosus. Azobacter basically is the nitrogen-fixing organism. Moxarella basically leads to conjunctivitis. We talked about lacunata as the particular type of organism that leads to disease. And then this is a photograph of Legionella. Again, you should be able to identify these organisms as well. Legionella, found in streams, water pipes, cooling towers. Again, this is dates back to 1976, where the group of convention goers met at a convention hall in Philadelphia and led to disease. And then Coxiella burnetii is the major one that leads to Q fever, transmitted via aerosol and or through milk. Vibrios are important as well. These, the morphology of these organisms, the bacteria, the genus Vibrio, usually slightly curved, kind of what the name implies. Important pathogens, Vibrio cholera. You may have heard of cholera before, but another organism that's important to be able to recognize. Parahemolyticus is the organism that causes gastroenteritis, transmitted mostly by the ingestion of shellfish. Can't talk today. Vibrio. These are some Vibriols. Vibrio cholera, again, causes cholera, and then also the parahemolyticus, which can cause gastroenteritis. E. coli, you're already familiar with, most common inhabitant of the human intestinal tract, familiar to most laboratory, those who work in laboratories. It's used as quickly identified through chemical laboratories that look at fecal matter. Again, very common in areas where you see a common mixture of sewage with the groundwater, which can lead to significant diseases. You also see it as the cause, one of the main causes of urinary tract infections as well. It also produces an enterotoxin, a particular class referred to as the O157 H7, causes serious foodborne diseases, and again, you've already looked at this in your laboratory studies. Salmonella, another organism that is important for you to know. Salmonella relates to typhoid fever. Most common, less serious gastrointestinal disease related to a microorganism. It's multiple serotypes or serovars of Salmonella. If we remember, we talked about the poultry industry being one of the most common areas where we'll see disease related to salmonella. Shigella, another organism that's important for you to know, bacillary dysentery or shigellosis, is basically responsible for causing traveler's diarrhea. It will infect only humans. And then the Klebsiella organism, Klebsiella pneumonia, is a microorganism that we see commonly as a cause of pneumonias in humans. Klebsiella pneumonia also exists, lives in the soil and in the water, fixes nitrogen from the air, major cause of respiratory disease. In Serratia marcescens, this produces a distinctive pigment, a red pigment. We can see this as well in the urinary and respiratory tract in hospitalized patients. Serratia marcescens typically will be one of the main sources of nosocomial infections that we'll see in hospitals. So you should be familiar with Serratia marcescens. Proteus organisms, common in the urinary tract. We also see it in wounds, but very common in urinary tracts, urinary tract infections. Grows on agar. Yersinia pestis. Yersinia pestis is a common cause of plague. This is another one of the diseases that's studied in bioterrorism. Again, the cause of plague, anything that can infect a large number of people in short periods of time, that can cause prolonged illnesses, may not cause death, but certainly incapacitates a significant portion of the population. So Yersinia pestis, which causes the plagues. And this is just a review list of the organisms that particularly that we look at that cause particular diseases. I focused you on Enterobacter, Escherichia coli. You've already studied Klebsiella, Proteus, Salmonella, Serratia, Shigella, and Yersinia pestis.